So the title of our CDE was for Biospecimens and Biomarkers, specifically for coma and disorders of consciousness. And the focus was essentially identifying laboratory biomarkers that can help with predicting both awakening as well as recovery from coma and disorders of consciousness in various disease states, with the goal specifically, for harmonizing future research in this area. The DOC, or Disorders of Consciousness Common Data Elements Steering Committee, convened a working group by invitation that was chaired by Dr. Sherry Chow from the Northwestern University. And then the other team members included myself, Vishank Shah from Johns Hopkins, Dr. Holly Hinson from UCSF, Dr. Mike Resnick from University of Pittsburgh, Dr. Cecile Hahn, who was our pediatrics expert from the University of Toronto, and Dr. Sheila Alexander, as well as Dr. Jonathan Elmer, also from University of Pittsburgh. So essentially, the working group was brought together. The goal was to come up with these biospecimen biomarkers, CDEs, and we met for the first time in March 2020. And during this meeting, we sort of, understood what the goal was to basically go through the NIH and NINDS already defined CDs, as well as identify new CDs through a systematic literature search that would involve laboratory biospecimens as biomarkers in various disease states that are associated with coma and disorders of consciousness. So we first went through the NIH, NINDS website and identified common data elements that are defined for various known diseases that cause coma, for example, ischemic stroke, subarachnoid hemorrhage. And then through that review, we identified essentially different common data elements that were pertinent to coma and DOC. And then we collated them, removed duplicates, and then had a follow-up meeting where we essentially re-adjudicated them using the new definition of the CDs that we're going to talk about. And then subsequently, we also conducted a literature search using a pre-specified set of search terms on common search engines, for example, PubMed. And we defined coma as the GCS of less than 8, and then DOC as the GCS of less than equal to 12. And then depending on the disease state, because there may be some diseases that are more rare and some that are more common, we had specific sample size thresholds for each different disease state, and each team member volunteered for one specific area of their expertise. And then they identified the CDEs, did the first adjudication into the different categories of the NINDS CDEs. And then in our follow-up meeting, we sort of developed a group consensus whether we agreed with that adjudication or wanted to upgrade or demote a CDE. And then eventually, once the entire list was brought up together, this was sort of presented to the other working groups and then converted into a case report form, or a CRF, which was then made available for public opinion and is now what has been published. And so that was the entire process that we followed. So our overarching goal was essentially to define key methodological elements and relevant time points for biospecimen collection, and then provide core and basic biomarkers that can be easily implemented in disorders of consciousness studies, or a broad array of study designs and in various types of resource settings, regardless of type of biofluid or frequency of sampling. And so we essentially defined general core biomarkers as well as disease-specific CDEs for different diseases that included ischemic stroke, intracerebral hemorrhage, subarachnoid hemorrhage, traumatic brain injury, seizures and status epilepticus, toxic and metabolic diseases, neoplasms, delirium, as well as demyelinating diseases and encephalitis. And then through this process, we essentially identified two disease core CDEs, 9 basic CDEs, 24 supplemental CDEs, and 59 exploratory CDEs that were kind of put together in the case report form eventually. The general core according to the NINDS definition, is CDEs that are required for all types of studies, regardless of whether there is a neurological disease or not. And so our case report form didn't really include any general core CDEs, but that is what is published on the NIH NINDS website, and we kind of make a reference to that. The disease score CDEs were required for all disorders of consciousness studies, regardless of the type of neurological disease. So there were only two core CDEs that we could define, because since these are the ones that are absolutely required, and that basically included whether a CSF study was available or not, and whether a toxicology screen was available or not. Basic CDEs are also known as highly recommended. So these are that CDEs that are strongly recommended for all studies encompassing disorders of consciousness. These include common lab tests like blood glucose levels, ammonia levels, oxygen saturation, drug screen, leukocyte count, among others. And so there were a total of 9 basic CDEs. Supplemental CDEs are CDEs that are recommended for specific disorders of consciousness depending on the context and the goal of the study. So this included more uh, uh, commonly used laboratory specimens, but those that are not necessarily always required. For example, CSF studies, the troponin levels, hemoglobin, partial pressure of oxygen and carbon dioxide in the blood, and then other neurological injury biomarkers like GFAP and S100 beta that could also be specific for certain diseases. And then exploratory CDEs are those that can be considered for use in disorders of consciousness studies, but require further validation. So this one had the maximum number of CDEs, 59, and these included things like inflammatory markers such as IL-10, TNF, TNF-alpha, blood microparticles, and these were more sort of dependent on the disease that was being studied. And then there was a last category called key design elements, and this was very important as well because these are not really data elements, but they are methodological parameters that must be included in the study methodology for all studies looking at biospecimens and disorders of consciousness. So this included details, for example, the methodology of collection and storage of the specimen, processing of the specimen, date and time of collection, frequency of collection, etc. So these were the big categories and the types of CDs that are in the CRF. I think the one unique aspect of this entire effort, not just for our working group, but for all the different working groups that put together CDEs in different areas of coma and DOC research, was that they created multidisciplinary groups. So our group included representatives from neurology, critical care medicine, emergency medicine, nursing, as well as pediatrics, and we also had international collaboration. So our pediatrics expert, Dr. Cecile Han, was from Canada. And so this helped us create a CDE, KDE list that can be sort of applicable to many different types of settings and a broad patient population. So I think that was the highlight of this work. The CRFs that were created were initially presented at the 2022 annual NCS meeting, and then open after public review, and then have been disseminated and are freely distributed on an open source web sharing platform called Zenodo. The link is actually available in the manuscript that is published in the Neurocritical Care Journal, and we actually strongly encourage researchers in this area to download it and then refer to it, and then also if they have any feedback and any changes, to reach out to us through the Curing Coma Campaign email so that we can update and develop newer versions. As a part of this effort, we recognize that there is considerable work that has been done and that is being done, both nationally as well as internationally, in developing biomarkers and biospecimen for disorders of consciousness. But there is significant heterogeneity in the study design and the methodology and how the biospecimen biomarkers are reported, because of which we actually had to adjudicate majority of them to the experimental category and very few that we could include as core CDEs. And so I think this initiative by the NCS and the Curing Coma campaign to define these common data elements and key design elements is a very important one and a key step to ensure more homogeneity in in future research so that we can have interinstitutional and international collaboration and also allow for future meta-analyses to really define specific biomarkers that could predict awakening from coma and recovery from coma, and then also foster development of novel therapeutics in the future. So I think that's really the highlight of this work.